Everybody. So nice to see all of you. Welcome to this very special event on intercepting ovarian cancer. So my name is Matt Vanderheiden. I'm the director here at the Koch Institute, and I'm personally really excited for today's event, which is going to highlight the collaborative efforts of three of our outstanding faculty members, Angie Belcher, Sangeeta Bhatia, and Paula Hammond. And their efforts to use engineering solutions as a way to develop new prevention, early detection, and interception techniques for a truly horrible cancer, ovarian cancer, where we definitely need many of these new solutions. For those who are less familiar with the Koch Institute, one of the things that makes us different and special is we try to be a place that really tries to combine the fundamental study of the underlying biology of cancer with the engineering solutions that might help us either better understand questions we couldn't ask or to actually put into practice and solve some of the problems that are necessary to make progress. You know, as someone who is trained originally as a physician, I can tell you that I've learned a lot since my time at MIT. What I've learned is our engineers are amazing at doing things. If you give them a problem, they know how to solve it. And Paula, Angie, and Sangeeta really fit that exactly, and has actually been fantastic in past efforts to collaborate together. They have a long and rich history of doing amazing things together. And I'm super excited to see what they can do as they come together and try to tackle ovarian cancer. Just to introduce them, for those who may not know them, Angie Belcher is a professor of biological engineering. Until very recently, she was head of the Department of Biological Engineering here. And her research has really focused on how do we harness processes that nature has already taught us as a way to improve materials, get better devices, and you'll hear much more from her today. Sangeeta Bhatia, also a professor of biological engineering. I think you're also an EECS, is that correct? Okay, good. And she is also, like me, a trained physician. Sangeeta's research has really been focused on how to use nanotechnology as a way to address complex challenges. Early detection is some really great work that Sangeeta has done in the past and will contribute here. And Paula Hammond is the professor in the Department of Chemical Engineering, and she's also I think still the head, although not for long, because she took on a new, even more important leadership role here at MIT. But Paula, of course, is famous for her development of various nanoparticle solutions to better deliver drugs, deliver combo therapies, and some of her work has really made progress in cancer and other areas as well. And all three, of course, are members of the Koch Institute Marble Center for Cancer Nanomedicine. Now, in addition to hearing from them, I'm also quite excited that we are going to kick off the evening by hearing from an MIT alumnus, Parul Somani, who is going to share with us her personal story with cancer and why the early detection of ovarian cancer and better needs for improvements in women's health is something that is important to her. And I hope that you will find her story to be both powerful and sobering. I know I'm looking forward to it, but also very hopeful. So it will, I think, outline why we need to do more in this space. So Parul is a business leader. She's an award-winning patient advocate. She's a lived experience expert. She's been invited to share her experiences as a cancer survivor, a caregiver and a previvor with President Biden's Cancer Moonshot Initiative, as well as the World Economic Forum and other leading healthcare organizations. She was named the 2020 Raising Hopes honoree by the American Cancer Society and currently serves on the National Breast Cancer Roundtable and the Patient Family Advisory Council. Parul, thank you so much for coming here today. please come take the podium. We look forward to hearing from you.
Yeah. Thank you. It's been about over 23 years now since I first stepped on MIT's campus. It's definitely changed a lot. A lot of life has happened during that time for myself as well. You know, when I was a student in college, there's a lot of aspirations that I had for what life was going to be like and the titles that I was going to hold. I can firmly say that the titles of survivor, caregiver, and previvor were not at all on my radar. So my name is Parul Somani, and there's 3 stories that I'm going to share with you today, and it's about those 3 experiences that I've now had over the years. So in 2012, I was newly a mother of my first daughter, and I was just at a normal OBGYN appointment and was sharing my family history. You see, my mom had early onset breast cancer in her 30s. And no one for 30 years had ever mentioned to us the idea of getting genetic testing done. And I was lucky to have an OBGYN who learned of my family history and suggested the idea because, you know, if it runs in the family, there are actual measures that I can take for that breast cancer risk. And got the cheek swab done, and there's all sorts of insurance hurdles that I will spare you the details of, but thankfully did learn the information that I do carry a mutation in the BRCA1 gene. Now, this was back in 2012 before Angelina Jolie had gone public about genetic testing, about the BRCA or BRCA mutation. And so I really didn't necessarily fully know what I was dealing with. But I understood that I was genetically predispositioned to have a very high risk of both breast and ovarian cancer. So I was able to start getting mammograms and MRIs at an earlier age than I normally would have, which I'm very thankful for. But it was very soon after that I became pregnant with my second daughter. And so I was missing those mammograms and those MRIs. And I'm 38 weeks pregnant, thankfully later on in my pregnancy. I'm actually in a doctor's room, leaning back on a table for an ultrasound for the baby. And I feel it's like lump in this itch in my chest, and I feel a pea-sized mass. Now, a couple of years earlier, when I had learned about my genetic risk, I literally Googled, like, what am I actually looking for in a self-exam? And it was described as a hard pea-sized mass. So immediately paranoid for many reasons, actually went straight to my OBGYN with no appointment there, shared what I was feeling. And I was told it's probably nothing to worry about because I'm 31 years old and I'm pregnant. So it's probably a clogged milk duct. And it was really only because we knew of my genetic risk that we insisted on an ultrasound and biopsy. And so given that background, she agreed to put in a referral. But I never made it to that appointment because 2 days later, my water broke and my second daughter arrived 2 weeks early via C-section. Now, if that time wasn't crazy enough, just recovering from a C-section, having a newborn baby and a toddler in the background, my daughter was actually born with some complications. And she stopped breathing soon after delivery and had to be intubated. We had to be ambulance transported to a neighboring hospital so that she could be in an acute NICU. We truly had every reason to dismiss the lump and believe that it was a clogged milk duct. In fact, after delivery, after my milk came in, I actually couldn't feel that lump anymore. It would have been a year of nursing before I would have felt that lump again. Thankfully, we knew of my risk. We were paranoid enough. We advocated for ourselves, and we got an appointment made for an ultrasound and biopsy. My husband wheeled me from the NICU, because I still couldn't walk from my C-section, to the breast clinic. Even there, the breast surgeon doing the exam, doing the biopsy, said that the mass feels soft, probably a clogged milk duct, and nothing to worry about. So we went back to the NICU, and thankfully all the tests came out clear for my baby. She is now thriving, and we were able to be discharged and go home. And it was that weekend, the weekend of Father's Day. I'm on a Saturday, literally sitting on my bed, holding my newborn baby, and I get the call. And when a breast surgeon calls you on a Saturday, you kind of know it's not going to be the best news. And sure enough, I answer, and she says, "I'm sorry, but the mass was malignant after all." And that after all will always stick with me because no one thought it was anything, given my age and given my pregnancy. So I had stage 2 triple negative breast cancer, and that was followed by 10 rounds of chemotherapy and 4 surgeries. But I'm so thankful for having learned about my genetic risk and enabling that information to help me detect my breast cancer at an earlier and more treatable stage. And I became a survivor. In fact, this past October was my 9-year remission anniversary. Thank you. But as we know with BRCA mutations, it's not just the breast cancer risk that remains high. It's the ovarian cancer risk that is high as well. Now, I shared how my mom had early onset breast cancer in her 30s. No one had ever suggested she get genetic testing done. I was the first in my family, the first to learn that I carry a genetic mutation, which means clearly it came from a parent. Now, we assumed it had come from my mom, but it took time for my mom to be convinced to get the testing done, because whether it's just personally or culturally, there was guilt that she had given it to me. And it took time to get her to get that testing done. And sure enough, when we did, it confirmed that she too carries the BRCA1 mutation, which means she too has a high risk for breast and ovarian cancer. Now with breast cancer, she had been treated in the past, and so she had a screening plan underway. There was mammograms, there was MRIs that she was getting. But it was the ovarian cancer risk that was simply not being managed and not being even looked at. I had become aware of the preventive measures that you can take in terms of the, when it comes to screening, it's a loose term, but like there's blood tests and there's, ultrasounds that you can get. But the preventive surgeries were really the options on the table to eliminate that risk. When I first raised the idea to my mother about this preventive surgery, it, as you can imagine, was not received very fondly. It took me a year. I spent a year giving my mom PowerPoint presentations and I was an ex-consultant, so I know my PowerPoint presentations, okay? This is how your risk increases with age when you have this mutation, and how it decreases if you get this preventive surgery done. And it took a year. Finally, my parents were convinced. She's postmenopausal, and this could give us the peace of mind that we were looking for. So my parents were living in Washington State at the time. They fly down to California where I live. They met with my care team, and in 2018, in June of 2018, she agrees to get this preventive surgery done. Before going in for the surgery, they do the CA-125 blood test, totally normal levels. They do CT scans, they do physical exams, all normal, all clear, and she has what is a preventive surgery that we think has gone completely successfully. But standard protocol for doing a surgery like this is that you also then biopsy the removed organs. And we learned that the biopsies showed cancer had already been growing in the tubes. We miraculously caught it at such an early stage simply because we had chosen to go in for this preventive surgery. It didn't show up in the CA-125 blood test. It didn't show up in the CT scans or any other exams. And the biopsies also showed that the cancer was so high grade that had we waited until the end of the summer, just a few months, it would have been spread all over her belly. Even by that point, she did in fact still had to get further surgery and had to go in for a few rounds of chemotherapy, but she now just celebrated her 5-year remission anniversary. I've experienced being a survivor, and I've experienced being a caregiver, advocating for my mom and quarterbacking her health. But then I still carry that ovarian cancer risk as well, right? So I was 31 when I went through my breast cancer diagnosis, and I had become aware that that risk for ovarian cancer really increases after 40. So I had mentally prepared myself that I am willing to do the prophylactic removal of the tubes and ovaries at 40 so that I can also have that peace of mind. And in the meantime, I would be doing what screening is available for ovarian cancer, which is those CA-125 blood tests, the pelvic ultrasound. Now, what happened during COVID was that I was still going for those blood tests, but now my levels were increasing. So my CA-125 levels for 2 years started increasing. Now I was still in my mid to late 30s at this time. There was concern that they were increasing, and there was concern that now my mother had already been diagnosed with ovarian cancer. And so it clearly was the type of genetic mutation that could in fact manifest into a cancer for me and my family. And so the care team was now immediately concerned and felt that what would have been a preventive prophylactic removal of my tubes and ovaries now needs to be a diagnostic surgery. It now needs to happen earlier, when I was 39 years old. Now, logistically, having a surgery like that at 39 when you already thought you were going to be getting it at 40 doesn't seem like a big deal, right? I was like, okay, I was going to get it anyway, a year earlier, not such a big deal. But when it goes from being a choice that you are making, which is empowering, that you are taking control of your health, to a diagnostic surgery where there's an urgency and you don't have a choice, the psychology around that is very different. And even though I had had 2 children by that time, even though I knew I shouldn't be having more children because of the breast cancer risk and so forth, there's a whole level of grief that comes from having surgically induced menopause at such a young age. But there was no other choice. I could either not go through the surgery and just hope that it wasn't ovarian cancer, or I could go through this surgery, they could biopsy the organs and identify what it is. So at age 39, I went in for surgery, root tubes, ovaries, and full hysterectomy. And it turned out that thankfully it was not ovarian cancer, but it was benign fibroids that were growing in the uterus. And those fibroids could have been left there, no problem, and there would have been no impact on my health. But because they had led to a rising of my CA-125 levels, I had to go through a diagnostic surgery, a removal of my reproductive organs, and surgically induced menopause at age 39. Now I can stand here as a previvor, right? Someone who's taken that preventive action to avoid a cancer diagnosis, and be thankful because I have experienced what it's like to actually receive the diagnosis, and I can just be thankful for not having experienced that. But think of all of the women out there who may still be in their early 30s, may not even be, having had any of their children yet, and may have a benign fibroid. And for them to have their only option be a removal of their tubes and ovaries so that they can be biopsied, a removal of their ability to have children naturally is devastating. So the reason I stand here today and I share these 3 stories with you is, there's so many pain points in my entire journey as a survivor. I needed to advocate for myself. I easily could have dismissed my lump for breast cancer because I was being told it was nothing to worry about. I was being told it was a clogged milk duct. And it was self-advocacy that saved my life. The screening existed, but it still required patient empowerment to get my early detection. And when it came to the ovarian cancer, the number of people who just don't even have access to their genetic information and know that they might be at a high risk is frankly unacceptable given the decline in cost for genetic testing. Everyone should have access to this actionable information and the choice of what to do with that information. So at least they can advocate for themselves. And then when it comes to, if you do think that you're at high risk, not having access to screening methods that are not so invasive, not jeopardizing our fertility is also just unacceptable. And the fact that the majority of ovarian cancers are only caught at a late stage when it's no longer curable and effectively treatable is honestly a travesty. And so I stand here today because I hope that by sharing my story and my family's journey, that it sheds some light on the pain points that exist, but that through the presentations we're going to hear from our faculty members today, also all of the great research that's being done to address that, so we can avoid people holding these titles in their future. Thank you. Now we'll have Angie kick off the faculty presentations. Thank you.
Thank you, Parul. That was, I'm really choked up. That was so moving. And I'm so sorry that, that you went through that. But you're a hero. You're a hero to your mom. You saved your mom's life. And you're a hero to people to teach about the experience and to help educate people about testing, about self-efficacy. And it's just, it's an honor to be here with you today. Let's see, am I in charge of this myself? So I'm going to talk about my passion for trying to solve problems around ovarian cancer, trying to solve it early, trying to come up with screening technologies so that women don't have to have these these same kinds of experiences. Okay. So together we can intercept ovarian cancer, and it's going to take passion, innovation, collaboration, compassion, dedication, and tenacity for all the reasons that you just heard about today. I'm going to take a small transition here to tell you about a problem that was solved recently using these same kinds of approaches. And so bear with me for just a second. So you might have heard recently that there was a rocket that landed on an asteroid. And that rocket landed on the asteroid and recently in September brought back samples to Earth. Okay, so there was a rocket from Earth that traveled 63 million miles to this asteroid called Bennu. This asteroid is traveling 63,000 miles per hour, and it's relatively small. It's about 1,600 feet across. And so NASA in 2020 landed on this moving asteroid 63 million miles from Earth. I think this goes like this. Orbited this asteroid, took high resolution pictures, infrared pictures, LiDAR pictures, measured the elements, measured the different minerals on it, took pictures, decided where to land, landed on this asteroid for 6 seconds, picked up 20 grams of rock, and then traveled back 63 million miles to Earth and dropped it off. So basically, it did a biopsy on this asteroid. This is an amazing, incredible feat. And I should say, anyone who knows me knows I collect rocks. And 5 years ago for my birthday, my family gave me a piece of meteor, which I cherish. So I get how incredible that is to collect these rocks. But there's actually a movie. There's a movie 63 million miles away of this probe picking up those rocks. And it cost about $1.16 billion. I'm not complaining about it. That was a great mission. That was amazing. And it was an amazing team that was able to do that. And there is a chance, 1 in 2,700 chance, that asteroid will hit the Earth in 2182. So it's also a way of understanding how to move asteroids away from the Earth. But yet, in 2023, we still have no accurate way of diagnosing ovarian cancer at early stages. We can go 63 miles away, take high resolution pictures, but we can't go 6 inches inside of a woman's body and measure her fallopian tubes, which are stationary. They're not moving at 63,000 miles per hour, and find 1 millimeter sized tumors and read it out. I think it took 18 minutes to get data back from the asteroid. And how long do you have to wait for your MRI or your CT scan or something like that? So I think the technology is doable. So what's hard about it? Well, one of the things that's really hard about it is we've been looking at the wrong place for 30 years. We've been looking at the wrong asteroid. So we now know that that 85% of ovarian cancer comes from the fallopian tube. It doesn't come from the ovaries. We've been looking in the wrong spot. Ovarian cancer comes at it's difficult to diagnose because it comes at the same time at menopause many times. The fallopian tubes are deep inside the body, not as far as an asteroid. The new imaging, people say new imaging can be expensive. Overdiagnosis could be a problem. Well, we know that's not really a problem right now. Ovarian cancer symptoms are amorphous. Fallopian tubes are not visible by MRI, CT, or ultrasound. That's true. They're kind of considered invisible. And then this is one that was actually told to me. I put it in a nice way. Engineers don't understand cancer well enough to solve this problem. I think that we can't accept no for an answer. I think we say no. I think we say this is a solvable problem. And so the chances of a woman getting ovarian cancer during her lifetime is 1 in 78. For BRCA1 positive women, it's 46% in her lifetime, or 27% for BRCA2 positive. The 5-year survival rate for stage 3 or stage 4 is 39 or 17%, has not improved in 20 years. 70% of ovarian cancer cases are diagnosed at stage 3 or stage 4. And one woman is diagnosed at stage 3 and stage 4 every 3 minutes. And when I talked to my surgical collaborator this morning at 6 o'clock in the morning, which is the easiest time to talk to her, she said, remind them that's the same amount of time that it takes to make a bagel and butter it. Every one woman is diagnosed every time it takes to make a bagel. So here's screening. There were 2 really big papers published in clinical trials on screening. These were CA-125 analysis and ultrasound. And the largest study so far, 400,000 women in 2021, it was published that there is no effect on mortality by CA-125 and ultrasound screening. It did shift the staging, but it did not preserve women's ability to survive. Now, there's other study came out in 2022 that said women that had opportunistic self-injectomy that had their tubes taken out at a time of another surgery actually greatly reduced the number of cancers. And so at this stage of 2022, women that had their fallopian tubes taken out, of the 30,000, there were 0 cases of ovarian cancer. Now, since the study, additional 5 years out, there's been 1. We can talk about that later if you want. So we now know that the ovarian cancer really starts in the fallopian tubes. It starts in these precancer lesions and then spreads. They're called sticks. They're very early precancer cells, and they spread through this intrarenal space to become metastatic ovarian cancer. And so normally what happens, just like was described before, when you have your fallopian tubes taken out, you look at them with histology, pathology, but they look at the whole fallopian tube? No, they look at 1% of the fallopian tube. So if that 1% is not looked at, then you don't actually know if you have one of these precancer lesions. And when I first learned about this, talking to surgeons, they said, oh, wait a minute, that's a pretty easy problem to solve. And actually, if you ask me, Angie, would you rather send a probe to another galaxy, or at least to an asteroid, or develop a screen for ovarian cancer? It sounds a lot easier to develop a screen for ovarian cancer. So I'm going to choose that. So we've embraced this audacious goal of trying to develop an accessible detection and diagnosis technology for ovarian cancer in the next 5 to 10 years. I think that we can do it. I think that we can find a screen for ovarian cancer. And so what is it going to take? It's going to take engineers and physicians and patients and scientists and advocates all working together. We need a group of passionate people to solve this. But another really key important part is that we need to work with people who can enable out-of-the-box thinking. people like Breakthrough Cancer, and the Gray Foundation, the Marble Center, and the Koch Center, who said, we think this is important, we think it can be done, and are willing to support this kind of technology. And these are my great collaborators. We're all holding signs that say, I look like an engineer, trying to redefine what engineers look like. But together we're working on trying to address these parts. And so we're going to do it by looking optically. We're going to use something called hyperspectral imaging and non-invasive detection that's been used to look at plants and used in agriculture for a long time. We're going to shift the wavelength, and we're going to use light to look deep inside the body. You say, Angie, that's not possible. Well, this is a green laser here. I can put this green laser on my finger, and it doesn't go through. I can put this red laser on my finger, and it starts to go through. As you go further and further red, same as with the asteroid, you can go deeper inside, and you can start to do a diagnosis and develop new materials. I'm running out of time. That's overambitious. We developed actually 5 levels of imaging equipment that have never been funded, never been made before, to look deep inside of the body with light. We worked with surgeons at Mass General to develop technologies for mapping tiny tumors inside the body for surgical guided imaging. And this was not funded by the government. The government would not fund us to do this. This was all funded by the KI, all funded by the KI and the Marble Center and individual donors who believed that we could do this. We developed a system to look deep inside, in this case, it was a mouse, and find tiny tumors that are less than 1 millimeter in size and mark them for surgeons to remove. And we showed that it would increase life in mouse models. I want to show you one other example, a company that we founded called Cision Vision. We were also told that we couldn't do this. We couldn't develop a technology to find lymph nodes. And Jeremy Lee, a postdoc in my lab, did this. It was all funded by the Marble Center for Nano Cancer and the KI, where we used the same kind of technology to find lymph nodes that are now used in 6 hospitals, used in colon cancer diagnosis. It wouldn't have been able to be done if it wasn't 2 engineers trying to solve problems around cancer. Now what we're trying to do is develop a technology to find small lesions on fallopian tubes. It's a big team effort. This is funded by Breakthrough Cancer. This is a fallopian tube it's from a woman that had surgery. Fallopian tubes are not invisible, actually. If you find the right wavelength of light of excitation, if you find the right wavelength of light of collection, you can see them. They're not invisible. No big surprise. With that, we're trying to change early detection many, many years earlier. We want to do fertility, interception, and I'm going to leave it with that. Basically, it takes a team of dedicated people to try to change this disease. And I'm excited to be a part of it. Oh. My amazing colleague,
well, you can see what an inspiring place we inhabit here. Angie, that was a great talk. And Parul, thank you so much for sharing your story. Really brave and moving. Okay, so I'm going to tell you a little snippet in that context about what we're dreaming about. I love this conversation because, engineers actually are dreamers. We invent things that don't exist today. And there are all kinds of reasons that people would say it's impossible, and the clinical trials will be long, et cetera, that unless you start, you're never going to get there. So I'm going to tell you about our piece of the dream, working together with colleagues, which has to do with finding those early lesions that Angie just introduced to you. So let's see. In the fallopian tube, at the end of the fallopian tube, which we now know is the source of most ovarian cancers. So in a way, it's a misnomer, right? All these years we've been looking in the wrong place. At the end of the fallopian tube are these little finger-like projections. We call them fimbria. And when they start to accumulate mutations, you get what's called a stick lesion. And that can then progress, as Angie just said. And so what we're trying to do in our group is look for these lesions by making a smart ultrasound contrast agent. So just as Angie said, you can't see them right now with ultrasound. You can't see the fallopian tubes at all. And we're trying to make an ultrasound agent that would let these light up. And the idea then is that patients would have choices about whether they could preserve fertility or whether they have the surgery. And it really could just inform the whole sort of patient journey. So that's the idea. And the science of how it works, or how we hope it works, is that the signatures of these cells as they transform change. We know all cancers change from when they're born to as they grow and spread. And in particular, in my group, we've been interested in the things that these cancer cells make. They make enzymes that allow them to grow and spread. And they're a class of enzyme that they make, and they're called proteases. So for some years now, we've been making protease sensors. These are enzymes that chew up the surrounding scaffolding tissue as those cells grow and spread. And so we invented a sensor that, here I'll just show you a generic version of it. It's a blue nanoparticle, and here it's in the bloodstream. And we've designed the nanoparticle to sort of leak out into the tissue where you're looking for an incipient cancer. And these blue nanoparticles are sensitive to these yellow proteases that early cancers make. And they get activated. And the nanoparticles are activated in a way that these little reporter molecules come off, the little tinier blue nanoparticles. And what's nice about the tinier blue nanoparticles is that what we've learned in the nanotechnology field is that these actually can be filtered out by the kidney, because the kidney is a really smart nanofilter. It's a 5 nanometer filter. So if the parent particle that I put in is big, it goes into the incipient cancer environment, it senses the proteases that the cancer is making, and then it can give off a flurry of these reporters, then the body's own kidney can concentrate that signal into the urine. So the way this plays out is it's a shot, an injection, and then a urine test about an hour later. So this is a technology we've been working on for all kinds of things, but we looked at it for advanced ovarian cancer and made this version and compared it to the existing blood tests. So here's a version of that blue nanoparticle I showed you before, and we designed it to sense a particular protease that is comes up when cancer is spreading in the peritoneal cavity. And in this mouse model, what we did is look in the urine of the mouse. There's a whole conversation about how you make mice pee, but we won't talk about that today. The urine of the mouse. And you can see already in this model of late stage spreading ovarian cancer, in 2 weeks, you start to see a signal. And if you compare it in the mouse to the blood test, you can see it's about a week earlier than the earliest blood signal. And in this aggressive model in this mouse model, this corresponds to about 3 months in humans. So it's not yet as long as we'd like, but it's a little bit earlier than the blood test just on its own. So this is what we had started when Angie and Paula and I got together through Breakthrough Cancer, and I really learned for the first time about stick lesions. And we started thinking, oh, there's an earlier window in biology where we'd like to point these sensors at. And in particular, we'd like to not just have a urine signal, but we'd like to be able to visualize those stick lesions on the fallopian tube. So how can we kind of adapt this idea to making a smart contrast agent? So Susanna Elledge in the lab is working on this. And essentially what we've done is look at, this is a long list of, again, well, these are proteases that these stick lesions make. So we work with collaborators in hospitals like Pakistan and others. We profile them, we look at all the RNA molecules, and we can see that they make some proteases. And when they become high grade and spreading, like the other model that I showed you, they make many more. But even early on, there are some very distinctive proteases that these lesions are making that we can point our sensors at. So then we're designing an ultrasound agent, and this is how it works. So this is an ultrasound like the one Angie showed you, and essentially the message here is that the fallopian tube is invisible typically on ultrasound. There are contrast agents that some of you may have gotten if you get an MRI with contrast or a CT with contrast. So there are contrast agents for ultrasound. What they are these little gas-filled bubbles of gas-filled bubbles. And you can see here, this is not in a fallopian tube, but you can see this is an ultrasound image with a contrast agent. So our idea was essentially to make a protease-sensitive version of one of these contrast agents. So the idea is wherever the tissue is making this protease, wherever this one of these stick lesions, that part will light up with the ultrasound lesion. And we have some chemistry we're working on based on the way that we made the nanoparticle that I showed you before. And essentially what it is a microbubble that is not sticky until it encounters the protease that we designed it for. And when it encounters that protease, this little veil gets cut off and these factors get liberated, and now it can stick to those stick lesions and light them up on ultrasound. And what we hope it looks like is this. This is an image from my colleague, Mikhail Shapiro at Caltech. He's actually an MIT alum in a different cancer model, but you can see this is the kind of thing that you could see for contrast imaging by ultrasound. So we really are just getting started. Susanna in the lab has made a sticky activated version of these that are sticking in response to proteases in the lab. We're doing ultrasound imaging here at the KI, and we're hoping to be able to visualize these lesions that were invisible before, and really potentially contribute, as we're dreaming, to a change in the paradigm for patients on the options. So with that, I'll thank you all for your attention and call my colleague Paula up here.
Well, that was really very exciting. I have to say, I am always so honored to have the colleagues that I have. And Parul, hearing your story was just so inspiring. We need people like you in the front of the fight, because you're the ones who really inspire us and help us understand how important these problems are. Now, I'm going to talk a little bit about treatment as well as detection. I'll tell a story that takes us through treatment, and then how what we learn from that might be used to help us with detection. And so hopefully you'll stick with me. In fact, I'm going to be talking about making nanoparticles that are particularly sticky to ovarian cancer cells. Let's see, oops, moved a little too fast here. All right, so as we mentioned, one of the real issues here is late detection. Another one is that ovarian tumors are very poorly responsive to common immunotherapies. So a lot of the advances that we've seen with immunotherapies and checkpoint inhibitors have really not yielded anything in the treatment of ovarian cancers once they do progress. Yet, ovarian cancer is one of these cancers that, although it may respond early on, comes back, it recurs. And that recurrence is something that we would love to address using the immune response. So we've been looking a great deal at immunotherapies as a way to address cancer. Now, in our lab, we actually design nanoparticles that are essentially created using charge. So we start with a negatively charged nanoparticle, and we know that negative and positive attract. So we can actually modify this nanoparticle. The nanoparticle itself is interesting. For example, this is a liposome that's shown here, and it can carry a range of different cargos, small molecule drugs, inhibitors, or RNA. However, we can layer that with a positively charged nanoparticle, and that positive charge allows us in certain cases to get in and out of certain parts of the cell once we enter. But the most important thing for us is the sticky part, and that's the second layer. That is this negatively charged polymer that we absorb onto the surfaces of these nanoparticles. They can be a range of different polymers. Some of them are familiar to us. Hyaluronic acid, we hear about in ads. We can actually plump tissue using, for example. Another one is a very common polypeptide, polyglutamic acid. These are native to the body, and yet they can have compelling interactions with cells. And it's those interactions that we try to milk to be able to find which ones will have a particular stickiness to ovarian cancer cells and not other cells. And in doing this work, what we found is that we can indeed make these systems fairly sticky. For example, we found two formulations. One is our hyaluronic acid one our polyglutamic acid one. They both really stick well to ovarian cancer cells over other healthy cells when we deliver them into the abdominal cavity. What we find is that they actually act differently. The first one causes these nanoparticles to go inside the cell and ultimately release their cargo to the inside of the cell. The second one actually sticks to the outside of the tumor cell, and what we release goes to the neighbors of those ovarian cancer cells. And that's actually meaningful for the immune response that we want. This is just giving you an idea of what happens if we inject our nanoparticles into the abdominal cavity. Here we have the tumor, which is designed to glow in this case. And we find that the signal that we get from the nanoparticle is overlaid very nicely with the tumor. And we can find the same thing with metastases, that we get that same faithfulness, that these nanoparticles find the mets as well as the primary tumor when we inject. So the idea then is that we might be able to deliver something like a protein that excites the immune response. Because one of the real issues with ovarian cancer being poorly responsive to checkpoint inhibitors and other immunotherapies is that there aren't a lot of immune cells present. They tend to be immune cell poor. And for that reason, they're not big responders. So in our work with Darryl Urban, we actually looked at the release of a cytokine. A cytokine is a protein that causes or induces an immune response. And it's a very potent agent. But it can actually get NK cells and T cells, these cells that fight the tumor, activated against the tumor. The problem is you really, really want to deliver something like this protein only in the tumor. If it gets into the rest of our bloodstream, it can actually cause a negative response. So a nanoparticle is a great way to deliver this system. And the idea was that we might be able to take our nanoparticle that sits on the outsides of cells and expose the neighbors to this cytokine. This cytokine has a tendency to cause immune cells to infiltrate in, kind of signals them in, and they begin to act on that tumor. So that's really the general idea, is that we attach the protein to our nanoparticle, we coat it with our sticky coating, we get it into the tumor, and we can see results. For example, we can see that the tumor sizes get smaller. If we look at the blue and the red versus the untreated tumor. And we can also see that the survival is much longer. So in this mouse model of a very aggressive form of ovarian cancer, we can see the non-treated animals. We can see what happens if we just introduce a checkpoint inhibitor. And then when we look at our different formulations, we have a formulation that when it's combined with a checkpoint inhibitor, gives us complete survival out to 200 days in this model. We're really excited about that because in mouse life, this is actually quite a long survivability. But the idea is that this is a very modular platform. And this is where I really enjoy the chance to collaborate with my colleagues, play with the neighbors, and actually do some really exciting things. This is some earlier work with Sangeeta, in which we were examining whether or not we might be able to combine some of the theranostic properties of her system with our ability to incorporate siRNA into our layers. And here you can see a simple example in which we're using the reporter concept that Sangeeta has developed on the surfaces of these sticky nanoparticles, which can then be delivered to tumors. And we can see that we get the response, the dark gray and black compared to the normalized tumor. In all of these cases, indicating a signal for the presence of tumor. But we can also see that the siRNA is active and silencing the gene. Here you can look at this blue bar compared to the gray bar. This is the beginning of a concept that we hope to evolve so that we can deliver therapies at the same time monitor what's going on during the treatment. see if we can actually decrease the activity of the tumor and understand what's going on. But of course, we want to go back to that idea of detection. And what if we can apply the stickiness to a system that allows us to see the ovarian tumors? Well, in this case, we have already done some early work with Angie's lab and used the ability to coat her nanoparticles that have these really unique near IR properties to be able to detect ovarian cancer. In all of those other images, we were looking at special luminescence for the tumor or pulling the tumor out and looking at it. Here, we're looking straight through a mouse, and we're seeing that these sticky nanoparticles that are sort of sticky antiparticles in that sense are actually able to allow us to visualize ovarian tumors. And this is actually some of her earlier systems. Now we have the opportunity to work with some of her highly evolved systems and apply the same kinds of technologies. But Angie and I have also been working together on making sticky proteins. And in this case, we've been working on a protein that can excite the immune system, but also stick to the checkpoint binding sites on other cells. And in that process, allow us to have kind of a two-part way in which we can address tumors. And here you can see a couple of examples where we've extended the lifetime, again, for an extended time period. So hopefully I've given you an idea of how we can have fun with friends. This is my lab, and I'm very proud of all of the members of our Koch Institute. We really are a diverse group of people engaged in science to help people. So I'll stop there and invite our colleagues to come up, and we'll have our Q&A.
All right, well, to get started with a couple of questions, Angie, you had shared some of the challenges that research in ovarian cancer has faced over the years. If I think about my own journey, it's been, just even from 2012 learning I have a mutation, and then 2021 still having to have such an invasive surgery, doesn't seem like there had been a lot that changed during that time. Which is consistent with what you said. And I'd love for you to expand a bit more on what has been some of the friction that has caused some of the delays in advancement in the ovarian cancer space.
So I think that's a great question. I think for one, there's been less financial focus on women's health, which I think definitely needs to change. I think that another kind of really important thing that we discovered was that that most of the ovarian cancer comes from the fallopian tubes. So when the very large clinical trials were started with 400,000 women, it was kind of in the beginning or the middle of those large scale studies that they realized that they shouldn't be looking at the ovaries as much as the fallopian tubes. And so I feel so lucky right now. I feel like all the pieces are starting to come together, and the science, the engineering. I obviously wish it would have happened 20 years ago, but I feel like the timing is right. That we have the idea of opportunistic self-injectomy, women having their fallopian tubes coming out is now very accepted. Although we need it to be more accepted, we need insurance to pay for it. We need more genetic testing. But part of it was we were looking in the wrong place. Part of it is we need to develop new technologies. It wasn't very technology driven, and what you saw from all of us were the development of technologies. And I think that's it says a lot to push things forward.
Yeah, no, absolutely. As we think of advancements in technology, obviously artificial intelligence, machine learning have had significant advancements and surely have a role to play when it comes to cancer research. For all of you, how do you think about what needs to happen in order to be able to actually apply those technologies to the ovarian cancer space?
I think there's a lot of exciting opportunities. In fact, I mentioned the idea of finding these sticky nanoparticles. What it's really about is understanding what kinds of proteins or what kinds of glycans or other native molecules may have an interaction that will facilitate delivery, for example, to stick lesions. Being able to mine a much larger genetic library, especially as we gain access to important patient samples, which is happening in our collaborative space right now, is going to allow us to understand what are the possible targets. On top of that, being able to use machine learning and AI to help us understand design principles that are consistent with the cells that we're really trying to target is going to help us and so become an important tool.
Yeah, and maybe just to tie together the two concepts, I think, one thing that's been challenging in all of cancer has been, our reluctance to work on early detection and diagnostics. Therapeutics are obviously really important and we need better therapies, but early detection is hard. Trials are long, they have to be big, they're expensive, and one of my heroes, Bert Vogelstein at Hopkins, always says, like, if you win 85% of the time in diagnostics, people say, like, you still failed, right? which is very different. So it's just a very, very challenging field. And one of the opportunities is to improve the power of diagnostics, the predictive power with AI. So if you look historically, for example, at PSA, which in prostate cancer, which is a single biomarker where you sort of define a threshold. It used to be used for screening, and then people realized that it could lead to overdiagnosis, and then it got de-recommended, right? What we envision as a field in diagnostics, if you measure multiple things and you use machine learning to define a smarter threshold in the context of a given patient with a lot of data, that you can make much more accurate predictions. So I think that this moment in time for diagnostics, AI is a really powerful tool to make them have less false positives and less false negatives.
I have a little bit of a follow-up on that, Michelle. That was beautifully said. That in the case, you can think about getting a mammogram and how you read in the news a lot, and a lot of that technology was actually developed here, how you can use computer vision and AI to help assist in reading mammograms. So why can't you do that with ovarian cancer? Well, we don't actually have a way of imaging the fallopian tubes and finding these tiny tumors to begin with, and we need to find precancer lesions that are on the order of a millimeter in size. And as the technology is developed, and my group back here, 24 hours a day sometimes are imaging fallopian tubes directly from surgery, collecting so much data, we don't know what it means right now. But it's going to be very, as we look at hundreds of fallopian tubes, we're going to be able to use AI to do prediction, but we don't have any data to predict with yet. So that's going to come.
Cool. Yes, please. Well, we have a microphone, so we'll just bring that to you.
Thank you for your sessions. Can you talk a little bit about cell-free DNA and liquid biopsy? Because there are commercial products already available in the market. You can buy it from Grail, where I think the stage 1 sensitivity is around 35 to 40, stage 2 is around 45 to 50%. And as you probably know, there are billions of dollars from private capital going into Freenome, and there is one just out here in the street. And there are at least 10, 15, 20 companies trying to look at circulating tumor DNA and cell-free DNA for doing early detection. And so how does that fit in? Because if that's successful, it looks like it's going to be successful, it's just a blood draw and you can do early detection, right? So what are your thoughts about that?
Yeah, I could take that. Thank you for that question. So it is a very exciting space. And for those who aren't familiar, so the idea is that a tumor will shed cell tumor DNA, free DNA into the blood, and you can take a blood draw and you could detect a cancer early. You may not know where it is, but at least you could then track a patient for other studies. So it is a very exciting field. The early stage data, as you say, are not there yet. And I think we're all hopeful that they will get better. Actually, here at the Koch Institute, together with the Broad, some of us are working on particles that will improve ctDNA even further. As the tumor sheds DNA into the blood, it gets cleared by the body. It gets cleared by enzymes in the blood, and it gets taken out by the liver. And if you can inhibit those processes, we've shown together with Chris Love's group that you could increase ctDNA levels by 100-fold, which we think is actually really promising as a supplement to the liquid biopsy tools to make them better for early stage cancers. So I think it's great that the field is coming up commercially, but the problems aren't solved yet, and we're still going to need new tools to sort of achieve the dream.
I wanted to go back to the funding. You mentioned that you couldn't get public funding. Do you see this work, is there a path to additional public funding for ovarian cancer research? And obviously KI is doing great work, and a couple others were mentioned, but who else is sort of funding research actively in this space and making change?
Well, I mean, I'll just start, and I have to say, I love working in the KI. This is the most incredible place to work for lots of different reasons. One, you have amazing colleagues that all collaborate together. We have amazing graduate students and postdocs and research scientists. It is like working in like Disneyland. It's amazing. But the one thing that's so great about it, and Susan Hockfield, who's here, was president at the time, and this was her vision, along with Tyler Jacks, is that we have this funding for really crazy ideas that you can't go out and get government funding for yet. We have internal competition. The Marble Nano Medicine Center that Sangeeta leads, it's based about funding people, engineers mostly, engineers mostly, with interesting ideas to get things started. We built five levels of instruments that no one had ever built before, and it was all based on internal funding from donors and from the KI that we wouldn't have been able to build otherwise. Actually, the one company, Cision Vision, we were told that wasn't possible, and so we just did it. And now it's in 6 hospitals. And so engineers just roll up their sleeves and they do things. And so we're really fortunate at the KI also to be part of National Cancer Institute. So we do get funding for facilities and things like that. But my experience was that people weren't interested in developing new technologies for early detection. That there was more interest in later stage cancer, but you both can talk about it as well.
I would first of all agree with Angie. I think it's much more difficult to get early detection funding. And I think there's also different ways in which funding works. And in DOD, there's been some good funding for ovarian cancer. And so I appreciate that. NIH also, but I think because of
and Paula got a 5-year award from them, and she did a sabbatical, and she went to the Dean of Harvard just to focus her whole time and energy on it.
Yeah. Absolutely, and that was really what brought me deep into ovarian cancer. I do think that there are review processes that sometimes make new and more radical ideas more difficult to get funded by our traditional agencies, because there's some, I think, review culture which says, well, you need to show me that it's working now, to fund it. And of course, that's counter to the actual idea. And I think as a country, we can work on that, but it's
or that it'll change survival.
Exactly.
Right? You just have to build it and do the trial
to find out.
Right.
Exactly.
I should say that we've all been fortunate to be funded by both the government and foundations. We had mentioned Breakthrough Cancer, which is based on radical collaboration, very interested in ovarian cancer. We're also just launched a new Gray Foundation, a grant that we're working on with Kripa, who I saw around on here somewhere as well. And these are people that are passionate about solving a problem. There's usually a connection, a family connection, or the need to solve the problem quickly.
Thank you for the great series of inspiring talks. I had a question about animal models. Can you comment on how much primary ovarian cancer models are available? Because I feel like all of us tend to use the peritoneal metastasis model. And if we're limited to that, can you also comment on how translatable our findings in those metastasis model are to the primary models?
Who wants to take that one?
Yeah. I'll take a bit of a stab because in our lab we've looked at several models and are always hungry for more. And we have great collaborators like Bob Weinberg, who introduce us to models that allow us to look at different kinds of genetic mutations within cancer. There is still a challenge in terms of replicating the environment, especially the early stage, where we can actually begin to look at what's going on, the development of stick lesions, et cetera. I think that we will all benefit from more work on developing models that are better able to replicate some of the stages of human disease.
Yeah, I agree, and I'll just add that one, I think, relatively new tool is this idea of like a human on a chip. So a graduate student in my group, together with Angie, is working on like a fallopian tube on a chip. And that the idea is that you could actually recapitulate a human fallopian tube under the microscope and watch these stick lesions evolve. So I think, mouse models don't sit alone, and we're trying to sort of develop all kinds of models.
I think I don't really have a comment directly on that, but for our imaging technology, we've been really fortunate to get an IRB to work with whole human organs, and I think we've looked at seven. We've looked at 7 patients so far, and we're hoping to look at hundreds this year. That doesn't necessarily answer the question around looking at therapeutics, but what it will do is allow us to collect these stick lesions from human samples and start to grow them in organoids and organ on a chip, where we'll be able to test those. And once we get to there, of course, we're happy to share those with you.
Hi, I have a question for our first speaker, Parul. Thank you for sharing your personal story with us. You also mentioned that you were fortunate to have genetic testing done at an early stage. I was wondering if you could comment on the barriers to making this more widespread. Like, are those insurance billing related barriers, or is there an awareness problem among the general population? So that we can bring this window of detection earlier on. Thank you.
Thank you for the question. It's a bit of all of the above. So, back in 2012, at that time, the patent for genetic testing was owned by just one company. And so I had gotten genetic testing through Myriad, which was what was available at that time. That patent has since expired. And so testing is now available from a number of other companies. Historically, the cost for genetic testing was incredibly expensive. So when insurance companies decided what criteria needs to be met in order to cover the cost of genetic testing, they established pretty high criteria, meaning pretty extensive family history is needed. So at that time, when I had shared that my own mother had early onset breast cancer back then, that itself wasn't enough to qualify for genetics. So when I referenced insurance hurdles, insurance had actually initially denied me coverage, and I needed to be able to share that there were more family members who had it. So that in and of itself is a friction, because many people, just don't have those conversations with their family, don't know their family history. Maybe they're adopted, don't have access to their family history. So to be able to provide that information itself is a barrier. But the fact that even, so I think if your mom had early onset breast cancer, that now is enough in terms of guidelines to qualify for coverage, but it's still a pretty high barrier. And now you're having companies, because the cost has come down, but the insurance coverage just hasn't necessarily caught up. You do have a clinical grade genetic labs out there that are providing medically actionable genetic testing, and they have self-pay options that are, out-of-pocket expenses, but much less than they used to be. So now for $250, you can get a panel of not just a BRCA, 1 and 2 tests done, but a number of other genes that are also actionable in nature in terms of there is screening that you could do to get them. And so this is where now gets to the second point that you brought up around education, is that both amongst healthcare providers, there's very limited time in med school spent towards genetic testing. And so a lot of primary care providers, for example, OBGYNs, don't necessarily have the familiarity. And that's why I consider myself lucky to have an OBGYN who even suggested genetic testing to me. And then there's obviously the education amongst consumers and patients and caregivers to know to even ask for that. And the reality is, when people do think of genetic testing, they also just think of, okay, like if I have a family history, then I should think about it. But when it comes to BRCA mutations, there's studies from Mary Claire King out of University of Washington who discovered the BRCA1 gene that shows that 50 or over 50% of women who have a mutation in their BRCA gene don't actually have a family history of cancer. And so there's education to be done out there too, that family history alone isn't like the only time you should be considering genetic testing. I don't know if there's anything anyone would like to add, but those are some thoughts.
Perfect.
Yeah.
Other questions for any 4 of us?
Hi, everybody. I'm Karen Spada, and I work here at the Koch, and I am unfortunately the person who has to let you all know that we are out of time. But the good news is that we have a reception immediately following right out here in the lobby. And I think all of our speakers will be out there to answer any lingering questions and to have conversation with all of you. So I want to thank you all for attending. I want to thank Paula, Sangeeta, Angie, and especially Parul for their very inspiring talks. And thank you all. Please join us at the reception.
Periodic tables at the front, if you want a periodic table. We have extra MIT periodic tables. Take some for your daughters.
Oh, yeah, I will. Yes, absolutely.